I'm Dr. Clara Abraham from Yale University in New Haven, Connecticut. I was asked by the editors of Gastroenterology to discuss highlights of a review article coming out in Gastroenterology entitled Interactions Between the Host Innate Immune System and Microbes in Inflammatory Bowel Disease. This review is in collaboration with Dr. Ruslan Metsitov. The high density of microbes in the intestinal lumen leads to continuous communication between host cells and microbes. These interactions can be mutually beneficial or can have adverse effects and contribute to intestinal inflammation, including IBD. The intestinal immune system must, on the one hand, defend against pathogens and entry of excessive intestinal microbes, and on the other hand, allow tolerance to resident intestinal microbes. Different components of microbiota can have very different effects on the host. The composition of microbial communities can be influenced by a variety of factors, including diet, antibiotic therapy, environmental exposure to microorganisms, and neonatal exposures. Changing environmental influences have likely modified our intestinal microbial communities and contributed to the increased prevalence of IBD. Alterations in intestinal microbial colonization have been detected in IBD patients, but it is not known if these changes are primary in nature or secondary to the inflammatory environment. Certain bacteria can have large effects on the intestinal immune system, in part by altering the balance between regulatory and effector T cells. Examples include polysaccharide A from Bacteroides fragilis, which increases regulatory T cell populations, and segmented filamentous bacteria, which increase Th17 cells in intestinal tissues. Moreover, the colonization of the host with bacteria and the host's ability to properly recognize bacteria are critical for the development and function of the intestinal immune system. The immune system continuously monitors resident microbiota, and certain antimicrobial mechanisms are constitutively engaged to prevent overgrowth of colonizing microbes. Examples include the physical barrier formed by intestinal epithelial cells, constitutive production of antimicrobial proteins by Paneth cells, mucus production by goblet cells, and the control of microbial attachment and invasion by secretory IgA. For those bacteria that manage to penetrate the epithelial barrier, immune cells within the lamina propria and secondary lymphoid structures provide additional defense mechanisms. Importantly, when one defense pathway is deficient, other pathways can compensate. For example, mice deficient in signaling through bacterial receptor response pathways have increased penetration of microbes from the intestinal lumen. But these mice produce increased titers of functional antibodies against intestinal microbes that help compensate for the deficiency in innate immunity and allow them to better survive infection. The classical distinction between commensal and pathogenic microorganisms, although practically convenient, does not necessarily characterize the full spectrum of behaviors by these respective microorganisms towards the host. It is not understood how the intestinal immune system can discriminate between resident intestinal microbes with which it coexists and pathogens to which it must respond. Hypotheses include that pathogens make different modifications to conserved surface molecules such as lipopolysaccharide, or that the cell damage produced during pathogen invasion may lead to inflammatory responses. Genetic association studies in patients with IBD further highlight the importance of proper regulation in pathways contributing to the interactions between the host and microbes. Examples of this include polymorphisms in NOD2, a protein that senses peptidoglycan, which is a component of bacterial cell walls. Polymorphisms in genes involved in autophagy, which is an important mechanism for clearance of intracellular components, including invading microbes. And polymorphisms in genes contributing to the IL-23 Th17 cytokine pathway, a pathway important for responses to various microbes. In addition, changes to the host can, in turn, affect microbial composition. For example, diabetes and obesity and factors regulating immunity in the host can alter intestinal microbes. Animal studies have shown that when the altered microbes are then transferred into a new host, the microbiota can now transmit disease susceptibility to this host, thereby highlighting the dynamic crosstalk between host and microbes. Thank you.